The reactions in this lab occur on spot plates, which are porcelain or plastic plates that have these depressions called wells on them, sort of like potholes. They allow us to make qualitative observations with very little material and without causing a lot of waste. This is sometimes called microscale, but each well, each spot can hold 1 or 2 milliliters. It is important to generate an observation data sheet before coming to the lab, and throughout the lab it is important to make sure that each well is filled with the correct chemicals. This video shows the test for calcium ions, where we have the first column of wells filled with some drops of distilled water, which is our control. The second column of wells get filled with the same number of drops of a solution known to contain calcium ions, and the third column gets filled again with the same number of drops of an unknown solution. Because here we need to carry out 3 steps, we use an eyedropper and add 2 drops of each of the liquids into the first 3 wells of the appropriate column. After that, we describe the appearance of our 3 liquids in our data sheet. Step 2 involves adding 4 drops of what is abbreviated here as G2HA, an agent that bonds with calcium ions called glyoxyl bis 2-hydroxyanyl. Now across the row for step 2, each of the 3 different liquids is combined with 4 drops of this reagent. Then we describe the appearance for each of the liquids after step 2. Before moving on to step 3, the hydroxyanyl also has to be added to the 3rd row of your spot plate. When calcium ions are present and bond with this glyoxyl reagent, the solution must have an alkalinic or basic pH, and in order to make sure other positive ions do not interfere with our complex, a so-called masking agent, sodium cyanide, is added. So for step 3, we add 1 drop of the cyanide hydroxide solution across the 3rd row of our spot plate wells. Observe how similar the suspension in the middle well that is known to contain calcium ions and the suspension in the well with the unknown look. So that you can later draw good conclusions, it is important that you write up a good, clear description of your last step into your lab data sheet.